Right. Okay. Good. Thanks very much for staying this late, and I'll tell you a little bit about where I'm coming from. So, in my case, I was supported by RCSA through the called the Dialogue program, and this is the excerpt from the RCSA website, right? And the whole description is like a manifesto, in fact. But if you actually look at the underlying parts, and this idea was about the collaborations between biologists and physicists, and between theorists and experimentalists. And my understanding of the idea was that they sort of experimented to see whether we can build a culture in biology just like we are used to in physical sciences, for example. There's a good dialogue between coarse-grained big picture modeling and the experimental test. And if there's a discrepancy, and then there's a back and forth such that everyone gets better. Right? Let me tell you a little bit about the initial background, what I used to do before I joined the Dialogue program, which is like from 2010 to up until 3 years ago. So one of the things, the nature of research in our lab is actually very question-driven. But every now and then, if the kind of questions we want to answer require certain technology, but the technology doesn't exist, we have to essentially invent those things. And one of the examples that we are known for is the so-called the mother machines. This is what we invented about 10 years ago when I was at Harvard. And the idea is actually very simple. What you're looking at here on the top end is a very simple diagram or top view of the device where you have a long trench with a Y, and the thousands of microchannels are all attached to this main trench. So if you zoom in one of this, it looks like this. It is a narrow, long channel such that the rod-shaped E. coli cells can fit very snugly without being tethered to any surface and so on. But because they cannot bypass with each other, you can actually watch the single mother cells for a very long time. So if you plot the lineage tree, the family tree for a very long time, what you see at the end of this channel is the mother cells, and then, which becomes grandmother, great-grandmothers, and so on and so forth. So this kind of device worked pretty well, actually, from day one. And this is the E. coli cells growing very nicely in the mother machines, expressing yellow fluorescent proteins. So you can just watch individual cells for a very, very long time. This is a very high throughput. So when we did these experiments 10 years ago, it was actually quite remarkable because up until then, all the microbiologists we knew did experiments on the agarose pad. And then they just grew bacteria on our agar pad and watched them for several generations. And here we had a very simple and robust device where we were able to follow thousands of mother cells for hundreds of consecutive generations. And then the amount of data we generated were orders of magnitude than anyone had obtained until that time. So. And although this is the throughput of these experiments is actually 1 or 2 orders of magnitude higher than 10 years ago, still this is my very favorite movies after all these 10 years. I'm not going to basically continue these movies because there are parts where people always ask when something goes wrong there. One of the discoveries we've made here is known as the adder principles. And this is a very simple principle. In biology textbook, people love proteins and checkpoints. So for example, in the context of a cell cycle control, people believe that there are some molecular sensors which actively monitors how big the cell is. And if the cell size is different from the target size, then some feedback mechanism is activated and talks to the other molecular machineries to adjust its cell size. What we discovered based on this kind of mother machine experiments was something very different. E. coli is very smart, and then they actually came up with a very passive way of regulating cell size. So this is a cartoonized version of our discovery. It's known as the adder, as I said. Whether the cell is born large or small at the beginning, for whatever reasons, these cells don't care how big they are at the beginning. They simply care about adding some fixed amount of cell size, and that is enough to ensure size homeostasis. So no matter how big you are, they all are guaranteed to converge the target size using this kind of passive feedback-less mechanisms. So initially we thought that this is only for E. coli, but turns out that virtually most of their model organisms in bacteria we study follow the same principles, and by now it is clear that the yeast and up to mammalian cells also follow the same principles. So so the impact of this paper actually was become very pleasant from both my point of view and the people who worked in my labs. The problem with this is that in the end, we are supposed to study the mechanistic origin of these mechanisms, but we also knew around 5 years ago, within 3 or 4 years, we knew that we were going to solve these problems. The question is, we always wanted to understand the mechanistic origin of cell size control and homeostasis. What are you going to do? One way is that you retire and do something else in life. Another way is that you got to explore the new frontiers in your research agenda. So I knew that I had to find something different. So the Dialogue program was extremely useful because I met very nice and people who have a complementary skills and interest. And one of them was Laura Lechner at Northwestern University. And she is a cell biologist and also molecular biologist. She studies mitochondria using yeast as their model organism system. And in particular, she's very interested in mitochondria, which is labeled as a red here, which also interacts with the other organelles. I liked mitochondria from the beginning. So mitochondria, for example, as you all know, is a powerhouse of the eukaryotic cells. Okay? Because most of the ATP molecules are generated inside the mitochondria. And then from cell biological point of view, mitochondria are fascinating because they also talk to the other organelles and so on. And then when something goes wrong with the mitochondria from, say, physiological point of view, and then cells also become sick and eventually they age and they die and so on. But if you like evolution, it's a great example of a symbiosis because, as you know, the origin of a mitochondria is thought to be an archaea or bacteria. So think of this, look at the dimensions of it. And then this is a 5-micron bar, and this is something like 1 micron wide. And then it's very elongated in this case. So to me, they were just like a very long E. coli cells becoming filamentous and so on. So this was a very fascinating system. And someone like me, a physicist, who are coming from bacteria, probably thought that can enter this field. And then Laura really helped us to get started. What was just the first kind of problems we are interested in? Well, we wanted to be crazy, but not too crazy. So one of the things we asked was something that looks like E. coli. So imagine, for example, you are now yeast cells, and due to stochasticity, some yeast cells would be born with large amounts of mitochondria, but some less and so on. If the cells are born with the different amounts of mitochondria, how do actually yeast cells control or adjust the right amount, the content of mitochondria to go back to their target value? Right? Of course, there are two extreme cases. One case is, again, feedback-based system, which actively adjusts the synthesis rate of mitochondria and so on. And the other extreme case would be the passive way of doing it, just like how E. coli go back to their normal size following the passive adder principles and so on. So we thought that we can probably test this kind of hypothesis. It was clear that nothing like this was done in the literature. So I don't have the answer yet. It's actually work in progress. And because what we realized in the meantime was that to answer questions like this, first of all, we really should be able to, for example, estimate the amount of mitochondria, the amount of molecules inside. The kind of tools people have in cell biology is actually very primitive compared to what it can do in E. coli, because imaging is challenging, image analysis is challenging, and so on. So if you do the Z-stack of these images, and then you have to construct the 3D morphology of mitochondria, but mitochondria is also very dynamic, right? So everything actually is very, very challenging once we realize enter the eukaryotic cell biology. So what we end up doing, currently we are very actively working on, is that we realize we really need to figure out the morphology and the topology of mitochondrial connectivity in a very the right way. And this is how we actually end up doing. So if you look at the top left, you see these all contiguous green objects. This is a mitochondria using glucose as a carbon source. And the way we label this is that the photoconvertible proteins, which basically diffuse around inside the mitochondria matrix. So basically, whenever you see green, that means that there's a green photoconvertible protein is basically diffusing around. And Now we actually we also implemented the Dmd Mirror, which is basically the micromirrors that you have in your microprojectors. The way it works is that it has millions of micromirrors, and then we should shine the light, and you can deflect the specific mirror such that you can basically send the light or the proportion of light to the target positions of interest, for instance. So now what you're doing here is as follows. It's a method that we call a paintbrush. That let's say you are interested in some specific region of the mitochondria, and then you basically illuminate you send targeted illuminations and convert the color of the proteins from green to red. Then red proteins then propagate up until the point, basically they will travel as fast as they can go, as long as there's no obstacles inside the mitochondria. So now look at this one. There's only green at the beginning, and then we do the targeted illuminations, and then the color is converted from green to red, and then boom, it propagates very fast up to that point, and then it pauses, and then in the next 2 minutes, it actually goes even further. I don't know whether you can see this very well because it's red, but just take my word for it, and you should see the red color propagating starting from that converted point all the way up to that point. See? And if you look at the green labeled mitochondria, there's no surprise there because everything looks connected. But it looks like there's some disconnection here, and then a disconnection there, and so on. There's some pause of another 2 minutes, but it looked like a very disconnected, but eventually this red protein travels all the way to the end. So this is a kind of method. If you simply look at only one color, you cannot really tell, maybe you will be able to reconstruct the geometry of the mitochondria, but you cannot tell the topological connectivity and what's inside this mitochondria. But by using this kind of paintbrush method from the old Windows program, you basically touch one side and everything is topologically connected and the color propagates, right? And this way you can really map out the topological domain of individual mitochondria. So these are 3 very talented from undergrads to grad students and the former postdocs. They teamed up and they're essentially mapping out all the topological domains of the mitochondria at the network in live cells. So we're very excited about it because nothing like this really has been done. And the individual pieces all exist separately, and all we had to do was bring them together. And this is the kind of things we couldn't do without the support of these private foundations because all my grant was for E. coli studies. What the biology should look like 20 years from now? Okay? That's the kind of questions that we ask every now and then. And of course, I can't predict what the biology should look like in 20 years from now, right? Even 5 years from now, because it changes so fast. But based on the kind of explorations we're doing, we started discovering some of the things. What it should and shouldn't look like. If you go to Google and go to Wikipedia and simply type the keyword biologist, and you will end up with one Wikipedia page, and this is the first picture you will see on that Wikipedia page under the title biologist. It's a very hardworking the woman scientist working with either picking microcolonies or working with the micromanipulators and so on, right? And of course, there are many things that are also automated in biology, and this is one of the microscopes we have. It's a fully fledged inverted automated microscope. And how much does it cost? Well, if you want to buy really fully functional good microscope, you'd have to pay something like 200 to 300K. And we have something like 4 microscopes in our lab. So initially, when I had a lot of money, I bought 4 of those, right? So I spent like $1 million and for this kind of use. You might think I'm crazy. Yes, I agree with you. So is this a cost of automations? Maybe. But how about this? What are you looking on the left-hand side? This very iconic image, so everyone knows what this is. And how much do we pay for this? This is about $1,000. And where does this price difference come from? This is about more than 2 orders of magnitude larger than, more expensive than this. Is it because the technology that goes into microscope is 100 times more expensive? Absolutely. It's a scale of production. It is because the size of the market. The difference between the two can be summarized in one keyword. And the reason the left-hand side iPhone is much cheaper, although presumably the technology that goes into iPhone is probably 100 times more sophisticated, is that the left-hand side, your iPhones are consumables, whereas your microscopes are not. Okay? So this is the price you are paying for. So what kinds of technology do we really need in biology, for instance? You ask yourself, and let's start with this microscope. Okay? Are we still supposed to pay 200K to 300K 20 years from now to basically take some time-lapse images of yeast cells? I hope not. So here's a 3D printer on the left-hand side. So you may think, whenever I show this to people, oh yeah, I get it. You basically want to print parts of a microscope. Yeah, you want to build a microscope, right? And then actually not quite like that. The left-hand side is a 3D printer you can buy from Amazon, and then you'd pay something like $300, right? Now the reason I'm showing you this is that your 3D printers are at the level of consumables. 5 years ago, probably you'd have to pay $3,000 for this. 10 years ago, probably you'd have to pay $30,000 for this kind of 3D printers. In the meantime, the precision of these 3D printers, the precision of this the microscope head moves is way, way more impressive than 5, 10 years ago, where you had to pay a lot more. So one of the grad students hacked these 3D printers. Basically, he wanted to convert this 3D printer into a microscope stage. Okay, so this is Arduino. You pay $50, and then he's an engineer, so he reverse engineered, and then he hacked this 3D printer head, and then he moves this around. He can just basically control and move it like just like he's controlling the microscope. And the next step would be you bring other consumables, such as the lenses. Your lenses in the iPhone and your DSLR Sony camera are really equally as good as the kind of cameras you buy for your fancy microscopes. So there are a lot of consumables you can just bring in. Really, the only thing that is not at the consumable level for microscope is there's only one thing, which is your objective lens. You still have to pay a lot of money, but everything else, from stage to cameras and so on, you can build really much, much cheaper. In fact, we estimated it. In fact, I'm not the only one who is trying to do these kind of things. You can build a full-fledged microscope on the 5K. Really, nothing compromised from technical point of view. You can do exactly the same kind of experiments with this 5K microscope instead of using 200K and so on. So we thought that this is a great idea. So we talked to the NIH. God damn it, why they just don't get it. I was so frustrated. So I was very puzzled by this one. So if you like microscope, the so-called micromanager developed by Ron Vail's lab at UCSF and then Nico Stuman. So I went to talk to Nico in San Francisco, and I took a flight, and then I was very mad. I talked to Nico. Nico said, oh yeah, Suckjoon, micromanager is not supported by NIH either. They stopped doing that since 5 years. So if you want to do this kind of things and spread your technology, democratize the technology, you either need to build a company, or you need the money from the private foundations, or basically give up. So I'm putting this last slide as a kind of take-home message for Richard and so on. Think about it. If there's something you can, private foundation can make a difference, I think this is one area I think really one can do. And personally, I strongly believe that this DIY, do-it-yourself approach can really change a lot of things in biology. If you don't have money, don't worry. 20 years from now, I just cannot imagine we'll be bound by technologies. A lot of technicians will be replaced by robots, for example. I don't believe we'll be picking colonies 20 years from now. And this is one of the ways to reach the future. So this is my last message, and this is the usual slide. When I first moved to UCSD, and then I really had a lot of help from private foundations, and my career stage switched from junior faculty when I didn't know anything about life and in reality, and then I transitioned to the mid-career. And to be honest with you, the amount of money I got from this left group and the right group is like order of magnitude different, right? But then, perhaps because the way I see the world has changed, I really appreciated how much help I really got. So I didn't have to come here, but I really, in a way, volunteered. I wanted to tell you guys, we did the dialogue program, and therefore, to help me get started with this East project and some of the toy projects we had, I don't think I could actually have done this with the NIH R1 grants, for E. coli. So this is my story. Thank you very much.